Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening. I'm Yvonne Stapp, and I welcome you to Science for the Public's Contemporary Science Issues and Innovations. Tonight, it is our great pleasure to talk with an outstanding representative of modern innovation. Javier Fernández is a postdoctoral fellow in the lab of the Wyss Institute for Biologically Inspired Engineering at Harvard University, where he works with the founding director, Ingber. Dr. Fernández received his PhD in nanobiotechnology from the Institute for Bioengineering of Catalonia, University of Barcelona in Spain. Before joining the Wyss Institute, he was a postdoctoral fellow at MIT, where he developed a technology called micromasonry, a unique structure that he'll tell us some more about tonight. Our immediate interest is his more recent innovation called Shrilk. This material is a biodegradable and biocompatible replacement for plastic. Shrilk is very strong but light, and it can be molded into complex shapes and a wide range of flexibility things. It can be used for commercial products, but it is also of interest in medicine, specifically for tissue regeneration processes. For this development, Dr. Fernández has recently received the Bayer Early Excellence in Science Award, given to exceptional innovators who are shaping the future of environmentally friendly manufacturing. We will certainly be hearing a lot about Dr. Fernández in coming years, and it is an honor to hear him talk about his work tonight. Dr. Fernández, welcome.
Thank you, Yvonne. It's a pleasure to be here.
I'm going to start by just asking you about plastic, because we'll talk about Shrilk first. And we have a term now called the plastosphere. It's that serious. Can you give us that kind of a background that we can talk about your replacement for plastic?
Well, plastic is actually the last century is so-called the plastic age. So we have based, since the invention of the polymers, of the synthetic polymers, we have based our development in the production of plastic. And that has been great for several years until we realized that the overuse of plastic is causing a very big problem in terms of health and environment. And at the same time, we are facing a future problem about the source of this material. So very soon, we are going to have problems to produce the same amount of plastic at the same cost. And that is going to be a very big economical and social problem.
Okay, so it's both an environmental and health thing, but also it's coming down to it will be too expensive. It's been so cheap, and everything around you is plastic, but it has been damaging, and now it's reached its end point, basically.
Yeah. So we basically have two problems, one in the source of the material, one in the disposal of the material.
Okay, so then the next thing is to how to replace this, and you've come up with an extraordinary thing. You're getting a lot of high praise for this, and I'll ask you, how did you sort of figure out how to work on this? So you start with a problem. How do you work into it?
Well, the initial, I worked with this material before for my studies in cell surface interaction.
I see.
So I was using biological and biocompatible polymers for trying to give a signal to biological cells, to say the cells where they should go, how they should develop. So the thing is that I did my thesis in electronic engineering department, so I was working with materials that they were focused for electronic engineering, where you can manufacture a nanoscale very easily, but then when you go and put in contact with biological components, they don't like it, the biological components. So that was how that motivated my study in try to do the same thing, but with biocompatible materials. And sorry.
I was just going to say, this must be quite a challenge, or was quite a challenge at some
was
point.
kind of a big challenge, yeah. But was really fun too. So the thing is that one of the first materials that came across was the chitin. The chitin is very abundant and is very cheap, and also is in nature is used as forming nanostructures. So that was definitely like the material that we wanted to have. So after that, when I joined the Wyss Institute, the idea was, okay, you already know how to fabricate, manufacture with this material, but this material is really cheap. Can you extend this process that you have developed for producing nanotechnology to produce much larger stuff? And that is how Shrilk came out.
Can you step back here just for a minute? The chitin, spelled C-H-I-T-I-N. This is very abundant in various types of structures, like in insect wings and so on. Can you tell us anything about that structure in terms of its many uses, many many functions, many?
Well, chitin is all over the place. It's in insects, it's in crustaceans, it's in cells of most of the marine animals. So the thing is that it is used in very different ways, and also in most cases is that evolution that it came from different approach. So and you reach the same kind of structural material, and that is chitin. Chitin is a structural material. Is made to build strong structures. In the case of the insect, is particularly interesting because chitin is just mixed with proteins to make the whole insect skin. And that means like you make the wings, but you also do the articulation of the legs. So with exactly the same component, you can do very stiff structure as the wings, or you can do very flexible structures. And that is why it's so interesting, because it's a material that has really all the broad spectrum of mechanical characteristics with one single molecule.
Yes, I want to make sure that even with very stiff wings like beetles and transparent, very light wings like mosquitoes or something like that, it's the same kind of material. And but it's also in the joints and other structures, right? So it takes different forms, but it's this material. And did you say that did it evolve like the eye in different places at different times, but it all came to this?
Yeah, it's always fun in structural materials for that reason, because it's a long molecule that assembles in a very specific way and then makes like very strong structures.
In your field, were you just aware that this material was out there, or do you look at different materials? You say, I want to do X, and I look at these different things?
We look at very different materials, but as I said, is that chitin is making like nanostructures. For example, in the wings, in order for them to be hydrophobic or even to have optical properties. So all these properties are made or are tuned by the topography at the nanoscale of those structures. So we know that the material has the chemical or biological properties that we want, and we also knew that this material is able to fabricate and to manufacture at the scale that we want. So that is why it was always our first candidate.
I see. So it was a first candidate. You knew that. And then when you set about to develop this for things that we want to replace in products, and you had other uses for it as well, was that very different from, say, designing an insect wing?
At the beginning was really different. So the main thing is that this material came as a mistake, I will say, because what we were looking is that all this difference between the stiffness in different parts of the insect body, they depend on the amount of water of the material. So depending on how much water you have at molecular scale, so you can have like different mechanical properties and different stiffness. So the problem is that we were trying to do materials that they are as plastic, right? So you cannot consider that your material is as plastic if when it gets wet, it changes the mechanical properties.
Right.
So that was our first bad result. So then what I did, I am in a bio-inspired institute. So what I did is like try to look in the physiology of the insects. It's like how they do in order to prevent this. Right. How these insects do for prevent that the rain or like water is going to change their skin properties. And this was like just months inside the zoology department in Harvard, like learning about physiology of the insects and crustaceans. And the thing is that this material always is associated with a protein protein that is very similar to the proteins that made the silk. So what we decided was to take those proteins, we developed a procedure to extract them from the silk and try to combine them. And the result was again negative. So our material was really bad, and also this problem with the water is still being there, right? So what we did is that instead of going one step further, and instead of just using the components, try to reproduce how those components are arranged at the molecular scale. So in the insect skin, these components, they are made by layers. One layer of the chitin, one layer of the protein, one layer of the chitin, one layer of the protein. So we reproduce that structure. And then what we found is that the material coming out from there was like twice as strong as the strongest component, that in this case was the chitin. So, and that is how Shrilk came into to the world. And at the end, what we did with the water problem was exactly the same that the insect do. So it's like we coat it with wax coating. So insects, they have all this kind of chitin protein structure, all the skin, until the very last layer, the epicuticle, that is made of wax. And then like, that is how we solved the water problem, and we came out with a material that is like have the strength of some aluminum alloys.
So you really had to carefully imitate nature on this. Well, nature took millions of years to work this stuff out, obviously.
So we were copycats.
You were, yes, but it's very instructive, isn't it? That both structurally and then the chemical.
And you realize that there is no random reasons in nature. So it was for a reason, was in that kind of structure.
Exactly.
And then we found that we can reproduce that.
Right. Now, the product itself is called Shrilk. So it has these different components. There's one more here in the shrimp case of shell as well. Can you tell us the name, how the name came about then?
Well, the Shrilk came from trying to cut and put together the words shrimp, that is where we take the chitin, and silk, that is from where we take the protein.
Okay, and put it all together and call it Shrilk. Then, do you use the shrimp shells themselves and silk in order to produce this, then, and then make the fabric, the kind of material itself, right? Now, in the end product, you want to have different flexibility and stiffness and stuff. How did you accomplish that?
Well, we can tune that like a bit with the amount of protein that we are putting in the system. What we can also do is like when we are providing this wax coating, so we can decide at what point we do it, and then we can decide how which is the amount of water that we have in the system when we do that. Also, we can change molecular weights for changing those properties. So we have like a certain several ways to actually change
so that's not as hard as getting the material in the first place, creating the material, getting the understanding how to do the material was the big...
Well, I will say that was like a two-step process. First, we did the material, and then we understood the material. So it's like we have these results, so why we are having these results? And that's very rewarding, because on the other hand, doing this research in the insect skin is really hard because that's like a very small system. It's made in a shape, has a lot of other functions. So, but we have a system that we can control very well. So the next step was like to understand why these molecules, when they are put in this way, and they produce this kind of like outstanding strength, strong material. And then when you already know that, then you can start working in changing the properties that affect the material.
To get the different flexibility, I guess, is what the different other features about the thing. And so in terms of what you could produce, well, put some images also into the videotape when we do it, but these are products of this, right? And the beauty of this is that it's cheap to produce, and also that it's biodegradable. And can you tell us anything about these? Are they as strong as, say, that plastic bottle? Or
right now we have, let's say that when we produce this Shrilk, we got like a lot of feedback from the industry. We got like a feedback basically from people from the medical environment and people from the manufacturing environment. So of course the needs were really different. People from medical, they are like really focused into the biocompatibility, what happens when you put this in the body, how reliable is the whole process. So the people from manufacturing, they were like very worried about cost, for example. How do you manufacture this at larger scale? So one of the things that we did, one of the first decisions, was to try to like lower the cost and simplify the manufacturing of the material. And that is how we start doing these plastic caps. So for doing that, also we sacrifice part of the strength. So this material is much stronger than this one, but this one is stronger than that one.
Even though it's also, they're both very lightweight and stuff, but this is stronger in the end. While you're on that, and then we want to talk about the medical application of this, is there a problem with scale? Like, this is plastic. Lots of things are plastic that are all around us. Everything is plastic, and you would want to develop stuff. Is that a problem once you get to larger scale of?
Well, it's a problem for us now because we are working in a lab. So, and then there is, we are working at a scale that is very large for a lab. So, like fabricating plastic caps, and especially if you are controlling things that are happening at the molecular scale, is pretty hard. And also, is one of the let's say like the merits of our material is that we are able to make real things. So we are not working in the theoretical environment anymore. So we are working in the real environment. But then it's like we can fabricate like 10 cups, like 20 cups. So you need to scale this in order to fabricate like hundreds of cups. And that is like where is the challenge. But that is like, it's going to be like a completely different challenge.
I think so. That's more like the manufacturer's problem. They can work on that. I just meant, was there in principle anything, you know, to restrict it? It seems like not really.
Not really. I mean, like from the fishing industry, for example, like every year you are discarding around like 250,000 tons of chitin every year. So you are actually like start doing or manufacturing with chitin. What means is that you can first like lower the cost of the fishing industry because you don't need to dispose that material or dump it anywhere. So
it does get disposed normally, these shrimp shells, but this is
and then like of course what we expect is like when this process get farther, so you will be able to actually lower the cost.
Exactly, I understand that. And now then, so you could make any range of products. You're anticipating that, and then manufacturers have to take up the rest of that, and that's sort of outside the lab at that point probably. There is a very interesting use in medicine in this. Can you tell us what has been why this material is of great interest in medicine, because there were problems before?
Well, one of the things of this material is that all the components that we are using are extremely biocompatible. That means that when you are going to put it in the body, so your body is not going to react negatively, right? And also because the molecular characteristic of the chitin has is a cationic polymer. That means like has like a positive charge there. That's really a strength for biopolymers. And that's an advantage when you put in the body, because it's able to bind to most of the other components of your body. So the thing is that you have a material that is biocompatible, that can kind of have like nice binding properties, and also is strong. So that is why there was kind of like big attention from the medical environment.
I was going to say, isn't one of the problems of the synthetics that are used a lot, like valves or whatever, replaceable bits in us, that has been a problem, that it might cause some difficulty in the body, these other materials, these other synthetic things, and this one, you don't have that problem.
Well, we don't have the problem, and also we are targeting different kind of applications. In this case, our material degrades in the body. So what you expect is that this material is going to support the regeneration of your body, the healing of your body. But then when you have grow your own structure made of your own tissue, then these materials should disappear from your body.
But not in a negative way, not in a damaging, I guess that's what I mean. That's right. So I'm sure there's a lot of interest in the medical community about this. Is there anything similar to it in the medical community? Is this like a real one singular thing?
Well, I would say, like, in terms of strength, we are really good. So there are a lot of biomaterials out there that they are, the whole tissue engineering is based in being able to replace the function of one function of your body with a different material or a different component. So there is a lot of research, and there is amazing results in tissue engineering these days. So I
think it sounds like a very exciting area, actually.
It is, and so also it's very active. So it's like every week or every month you find someone doing or with a result that is really groundbreaking. So.
Right, especially with this nanoscale of that you can do so much now that, I don't know, 20 years ago, I'm not sure that you
and biology has advanced like
tremendously.
Yeah,
exactly. In terms of when you do a product like this, then are there ways for you to protect it? Do you get patents for this? This is less familiar to us in terms of developing products. So how does that work when you develop this, but you don't want everybody running off with it?
Well, it's more than running off. Is like our institute is an institute for translational science. So when this institute was found, so the founder made very clear the point that everything that is done should make a change in the society. It doesn't matter, it doesn't need to do economic profit, but should make a change, right? So one of the things that we realized and we know is that if I develop this, but I don't protect it, any company is going to follow up. So they are not, I need the companies right now, or we need the companies in order to make this material to go really to the market. So that is why we try to patent all this, and we patent actually. We have 4 or 5 different patents, different aspects of our technology. So, and that is because at some point we want someone to take over that and then go and do the next step and bring the society with something new and something useful. And they are not going to do it if they are not going to have economic profit, and they need to have a patent in order to have economic profit.
Exactly. But that sounds like it's a healthy kind of relationship. Anyway,
we have and also I would say that this institute is kind of an experiment in every sense, and it's really easy for us to work with the companies. They are really open to make every kind of agreements, and half of the people from the institute is focused in that, in relation with companies and development of products.
I see. Get back to the medical part again. You developed something else that's really very interesting. Is it micromasonry, the little cell thing? I wonder if you could tell us about that as well. And I wonder if you could also include something about that. I think there's a lot of work on scaffolding into building tissue, but yours is so close to practically print an organ. It sounded like, Could you tell us about that? And
there is actually other technologies that are based in 3D printing of organs, right? Or
your
things
liver.
that are similar to organs,
Yeah,
right? So when I was in the MIT, I developed this technique that is called a micromasonry. So that basically consists in that when you have cells, like biological cells, coming out from a human, from a patient, so what you need, first thing that you need to do is to break the joints between those cells.
Yeah.
So in order to be able to take them out, culture them, expand them, have more cells, and then you will take them and then you will use it for a cell therapy, right? So the problem is that there is an obvious step that the cells are breaking those connections and they just make layers. And that is what you have in the lab. So I came with this solution that is making or encapsulate the cells in small cubes, small cubes of a biopolymer, that they are made exactly in the same ways as you do electronic engineering. You shine some specific light on a material, and this material hardens, right? So this material is loaded with cells, and then, like, let's say, like, providing the right shadow on that material, you can do the shape that you want. If I make like only like one spot and then light through it, so I'm going to make a pillar through my material. If I do like a square, I'm going to do like a square pillar, right? So what I was doing were cubes loaded with cells, and then like make them freestanding. So like be able to actually take these cubes and then like put in wherever I want. And I did that with capillarity forces. So there is like the best example of this will be a sandcastle. So when you are doing a sandcastle and you have a very dry sand, you cannot do anything, right? So if you put like too much water, so it's like they just are wash out, right? But if you have the right amount of water, then it's sticky, and you can make a sandcastle. So this is because in between these sand grains, you have water that is keeping them together. And that is exactly what I did with these small cubes. So I provide them a way to stick together. And instead of using water, what I was using is this polymer that when you illuminate it, hardens. So you first make them to join each other, and then you illuminate them, and then you have the structure. And you can do it like 3D structures. We were doing cubes, we were doing spheres. What was kind of
I think that would have been tremendously exciting, because I'm not sure how much of this stuff has been done in 3D. Is that true? You know, it's been really complicated structures.
Well, the thing is that the people right now is doing 3D printer, right? Like the problem that you have is that the definition of that. And then the groundbreaking result that we have was that we were really doing the process very simple. Even if when I explain it sounds like a really complicated, at the end it's like you just need to illuminate twice and then have the right surface, and they self-assemble. So, and that's the potential of it, that at some point you should be able to put them in the right environment and they will form whatever they want, or whatever you want.
Yes.
Basically.
So where does that take us in terms of medicine, in terms of tissue development, in terms of like replacing organs and things like this?
Well, that's the basic thing. So it's like in tissue engineering, there is two main parts. One is to have the right cells, and that the cells do what you want. So if I want like cells for the liver, I want these cells to actually like be real liver cells, right? And make all the structure of the cells. And the second part is to provide these cells with the proper environment to make a liver. So what we were working was in this second part, like providing these cells with the right environment for making a liver, and then like be able to put them exactly in the place that they need to be. But then it's the second part. These cells need to grow, need to modify the material, need to make like the real liver.
And do you think this is all, it's going to come out right in the end here?
Tissue engineering is going to explode, so, and it's exploding, I will say.
Yes, I've seen different variations of what people are doing on this, but I thought this was really singular. This is really unique.
I would say that the difference is that we are not using a scaffolding and then trying to grow them and then like grow wherever they want. So we were like
this is more direct.
And also we are able to control who is your neighbor. In the body is really important. Who not only what kind of cells you are, is also which cells are surrounding me. Because if the cells that are surrounding me, they are different, maybe I decide to develop in something else, right?
Right.
So, and in this case, what we are really, we have like a very good control of who is at your side. And that's really important when trying to make the cells to develop in a specific direction.
This is really fascinating that you can do both the biological end and then product kinds of ends too.
Yeah, at the end it's like you have this kind of historical fields in science. When you are in science, you realize that everything is the same. So you cannot say like materials science is still here and biology is still here. So it's like everything has is under the same
This seems very much a trend, especially today. There was a time when people had their little tiny specialties, you know, or something, but it's out of the box now that everybody, as you said before, you seem to have to be learning, sometimes from the ground up, you know, a whole new field all the time. And you have to have, well, a lot of scientists have exactly that mentality. They enjoy that. That's a lot of fun.
Yeah.
But that is a lot of work, isn't it? But
well, I mean, it doesn't feel like work. I mean, if you are enjoying it. So, but yeah, definitely. I mean, I have been here like doing science since I was 17, right? So I'm professional since I was 22. So that means that it's a lot of years doing the same.
Yes.
Well, the same.
What would you like to be doing? I mean, what's like a next project, or what are you working on now?
Well, what I'm working now is like we have like many projects ongoing right now, but it's like, for example, we are producing an artificial skin right now that is really for burns, actually, for people with a really bad burn. Yeah. And that's been very rewarding. I'm very fun too. And that is like what I'm doing right now. In the future, I have this idea that is like, we can take materials take ideas from biology and make new materials, right? But I definitely think, and this is connected with the micromasonry too, is that there is potential to integrate all the biology components in the material. So we are just starting to explore the field of biological materials. But biological materials are much more than glass caps. Biological materials if they are done right and they are done like as nature does it, so they can regenerate, they can grow, they can adapt, they are alive, right? Yeah. I definitely think, and that is my research direction, and this is my vision, is that at some point all the stuff that we are doing will have these properties too, that you can really integrate biology into material science.
Yes, that seems to be very much the direction. I'd like to just ask very quickly about the skin thing. A lot of people are working on skin now, and that is evidently very complicated, but you're working on it with the micromasonry approach. Is that correct? And
no,
something different.
It's completely different.
Oh gosh.
So is that, and also I cannot like give too much detail obviously about this, but it's like it's the way we are developing is a spray basically, so that actually attached to the areas that has been like exposed, damaged, but then doesn't attach to those that are healthy, and then produce an antibacterial coating and artificial skin there. Right. And the good thing, or the let's say like the most impressive thing of the whole system is that you can paint it. Basically, it's like works as a spray, and you basically paint the person with the artificial skin.
That's quite amazing.
That's quite amazing. I mean, this is very experimental, but it's really
right. But that is several jumps beyond what I'm familiar with. Certainly, in that it seems like
people.
It's really,
I mean, this like painting a skin has been in the mind of scientists, and there are like several groups working in different approaches for exactly with the same ideas.
Right. This is very exciting, very rewarding too, because you're really helping people with a lot of the things that you are doing. What would you like to do if you could do anything now for the next 5 or 10 years? Is there anything before we back off that you would like to work on that you haven't worked on? And
I think I'm working on
what
I want to do.
Yeah, yes, so, you do get to do whatever you want to do. Basically.
Yeah, I mean, I will say that that's in my case, and in general in scientists, you are very free to decide on your direction. As you say, there is like certain point that you are just making a new, very characteristic line of research, and you are no more following a very specific field with a very specific name. And that's more or less, I mean, at some point you just look back and then you see like your path was like kind of weird, but
exactly,
it's what you are.
exactly. I expect you will get a lot of prizes down the road, and we'll see you again very famous, but you're certainly moving along in that line.
So.
Okay, I'm going to turn it over for let people talk to you, questions here, and I really appreciate your giving us this wonderful tale about these materials.
Hello.
Thank you for your nice presentation. I have a question about the physical properties of the Shrilk polymer. What is the melting point when you heat it? At what temperature does it melt? Can you put hot coffee in those cups, for example? And then also, at what temperature when you heat it does the polymer decompose?
So is that the polymers that we are using and most of the properties that it has is because it has a very well-defined crystal shape. So it's like it's very crystalline polymer. So that means that there is no obvious melting point. There is no glass transition temperature, there is no obvious melting point. So you will reach the carbonization of the polymer at 200 something centigrades. So, and Celsius, sorry. And that is when you like basically destroy your polymer.
Okay. So what temperature range can you process this to put it into a shape like a cup?
Well, we actually don't use temperature for putting, like making the shape of a cup. We play with water content in order to change the shapes of our material. And we use always, we move in a range of temperatures in order to like do the things that we do that are like maybe around like 60, 80 Celsius. We move in that range in order to tune up or vary a little bit the viscosity. But basically, we have an approach that is not common to the.
So the water content determines the viscosity.
The what?
The water content determines the viscosity.
Yes.
Okay, that's pretty cool. Okay.
You related your work to building with sand and how you need to have the right amount of water and the right amount of sand in order to make it work. As you said, it has much medical promise. If you were to build an organ or a liver, what kind of work does that take? Like, what kind of resources does that take?
Resources in terms of.
Well, just like what kind of work goes into that?
So what kind of work? So you mean like in terms of time, people like doing it, or?
Just like how would you go about doing that? What kind of working process is it? How do you go through it?
Okay. So basically what we do is like this is the micromasonry thing. So it's like it's a very fast process. So that is like why it's useful in this field, because basically also you do it in room conditions. So you cannot have biological cells a long time out of the like very controlled environment. So, but what we do is like we do these small cubes, and then we provide them with a surface. It's a surface that is, let's say in this case, will be a surface that is able to, like the sun is able to stick. So they can do like this same, the capillary breeze they can glue also with the water with your cubes. And then the difference with the sun is that the sun will break apart if you scratch it or something like that. But we have this second tool that is like if we illuminate that structure, it's going to keep that shape. So imagine that you go to the sea, you take all the sand, this wet sand, you put in your hand, and it's going to take the shape of your hand. And then you can make that harden and get like a stiff and then be solid. So that is what we do, like illuminating it. And that is how we build like large structure with this approach. We give them the specific shape, they arrange around the shape, and then we fix that shape.
Thank you.
In the post-consumer realm, how quickly does Shrilk break down? Is it suitable for your compost? Would there be a new number for your recycle bin? Will it contaminate other plastics if it gets recycled with them?
So it can be recycled, but as other plastics, you need to do like a specific process for this kind of plastic. You cannot mix them, because then at the end we'll end like with a kind of a material that you cannot manufacture anymore, right? So the good thing of this Shrilk is like is not biodegradable in the sense that you understand biodegradability in plastic these days. It's compostable. So it's like Shrilk is if you have enough humidity, if you have the bacteria, and those are like common bacteria, so it's going to become a compost. And you can do in your house, in your compost bin, and on the other hand, is like one of the major use of chitin these days, that is the main component of our constructions, is as fertilizer, because it has a lot of nitrogen. So the good thing is that you have passed from being a problem to the ground or to the soil to actually be enhancing the growing of plants in the ground. So it's fully compostable. So it's not biodegradable, as I need like a specific factory for that. Okay, my background is in fundamental physics, and I have a master in nanotechnology in Sweden. So my master was basically understanding and fabricating surface with nanotopography. And from that I moved to do a PhD in Barcelona, back in Spain, for a study how those nanosurfaces, they interact with biological cells. So from there, I moved to study nanomaterials, so how you fabricate with biological, natural materials, biocompatible, and give them like some kind of signals, topographic signals at the nanoscale. And then from there, I moved to the US and then to the MIT, and did some research there in tissue engineering, and now I'm doing research both in tissue engineering and also in sustainable materials. So the most satisfying part of my work, I guess, that is like to be always to have always a challenge. So that's science, right? So you always go to the lab and you don't know which is going to be your challenge, and you are learning every day. You are learning a lot, you are studying continuously, and that's really rewarding. You need to have a lot of that, and the thing is that many times you need to address a new field from scratch. So you can be afraid to learn like a new thing. So my background is fundamental physics. I have spent hours, days, months in the zoology department in Harvard studying old books. So it's always like you just are learning from scratch new things, and it's a never-ending process. I will say that the bad side of being a scientist is everything that is not related with science. As a scientist, you need to fill tons of paper that maybe they are not like continuously, or they are not directly related with your science. You need to apply for money. You need to figure out things that are more administrative rather than doing pure science. And that's like, let's say that's something that you need to do, but you don't want to do. I guess that this century is the people try to call it like the molecular age. So we are like learning how the nature do all their materials, and we have the tools to control materials at a molecular scale. So this is going to completely change how we fabricate, how we manufacture these days. And how I see how the field is going to develop is that in a very short time we will be able to make materials with very different levels of structuration of manufacture, and then from the nanoscale to the like real shape of the thing that you are fabricating.